Hello everyone, my name is Mauricio Bonilla Flores and today I will present my work that is entitled High Altitude Discovery, a new asexual ostracod from Namsor region, southern Tibetan Plateau. And this work was part of my doctoral thesis and is already submitted in the journal CEST. And here in this presentation I show you the female in the internal view and in the external view how is the characteristic of this new species. And it's important to mention that how the taxonomy is the important step because when we want to use this type of animals in environmental interpretations in the past, we needed this in good descriptions of taxonomy in order to have a robust interpretations in paleoenvironmental studies. Well, at the beginning, we have that the Tibetan Plateau is an important area because it's the highest altitude on the planet. It's a large area, and in this area also it has extreme temperatures and low precipitations, and it has many lakes, rivers, lagoons, and ponds. In this area, we have the second largest lake, Lake Namson. Is an endorheic brackish, and in this area, inhabit specialized aquatic fauna with resistant strategies. In this case, ostracods are animals with good sedimentary record since early Ordovician. It has this group. They have a good adaptation to extreme environments with sexual and asexual reproduction, and they are used in paleoenvironmental studies combined with prerequisite modern studies. Ostracods, they have important attributes. They have small size, they have widely distribution, they have short life cycle, they grow by molting, they have abundant and they have environmental sensitivity. An important thing is that they have a fossilized remains. In this case, the shells are conserved in sediments. And the first prerequisite is the ecology, is to relate species to environmental parameters. And the second one is the integrative taxonomy, the use of the description of morphology and also use molecular tools. And in the third one is laboratory cultures in order to describe on the ontogeny and to have good morphometric analysis. All of these prerequisites are used in these applications. For example, in changing species assemblage and ecological traits, infer environmental variables using training sets and transfer functions, and develop chemistry to infer climate and environmental conditions. Well, our objectives are three. The first one is describe heterocypress exodonta using k-morphological traits and co-gene data or the gene analysis, molecular analysis. Compare ball morphology of these three species, exodonta with the related congeners. And the last one is determine the geographic distributions of this species exodonta and the closely related species. Of the material and methods in the field where or some colleagues collect the samples and the sediment, and also they measure some immunological parameters. The samples were transported to this research station in Namso, and in the laboratory work at the Institute of Geosystem and Bioindication in Braunschweig in Germany, we weighed 1 gram of sediment and sieve 63 micrometers. and I separate the animals with soft parts and with hard parts. And the animals with soft parts, I made some dissections for the identification and the description, and also the use of the literature. And with the animals with hard parts, in this case only the use of the bulbs, we obtain some measures of the shells. Our results, at the first step, we compare the characteristic of the bulbs and the internal pores, and we observe with a comparison of this material that this species in the right valve, they have it has a prominent denticles in the borders, totally different to the other species. Also the pores, it has a turbine shape, and at the first step we compare this material with this other material from this other place in Taroko, and we observe that they have these species, they have these denticles are very small, and also the pores, for example, in incongruence is totally different. It has a turbine shape also, but it has a bristle in the center, and in the pore of the Heterocypresalina, it has a simple hole. That's at the first step in the comparison of the shells, but we also had material available from other places, for example, incongruence from Mexico City and Sarina from Braunschweig, Germany, and it was the same. And we recognize that these characteristics in the pores and the denticles in the borders is totally different. And in this until this point, we say that these characteristics with these characteristics we can differentiate the new species, but also use the with the soft part, we differentiate with the use and the description of these female genital lobes. We recognize that is totally different between incongruence and salina, and also from different places, and we use for the first time these characteristics in this work, because the genitalia of the females is less used in comparison with males, for example. And until this point, with these characteristics, we can say that it's a new species, but in order to be sure, we produced molecular analysis. That is, molecular analysis consists in several steps: genetic material instructions of this species, PCR amplification, sequencing, and data analysis. At the end, we produce these phylogenetic tree visualizations. This work, it was it was made with Dr. Ivana, in collaboration with Dr. Ivana in Hanyang University in Republic of Korea. In this molecular analysis, we produce this tree, and we can observe here in yellow how Heterocypresalina from Germany and Heterocypres from Tibet and incongruence from Mexico, they are in different branches, and that's the reason that in genetic data or in genetic, they are totally different species. Well, with the data of the records of this species, we can observe here that Ixodonta only is found in this area, in Tibetan Plateau, but incongruence and salina, they have a widely distribution. Our conclusions are the following: the shape of the bulbs and dentition work in differentiate the new species. The description of the internal pores in the genus were recorded for the first time, and the female genital lobes is different between these three species, and molecularly it's confirmed the separation of these species. And exodonta, Pterocypris exodonta has been recorded only in Tibetan Plateau, while incongruence and salina have a worldwide distribution. And that's the reason that we need to have different tools, in this case morphology and genetic data, for differentiate the species in order to have a robust inference in paleoenvironmental studies. Thank you so much.